speaker is Helena Key from Massachusetts Institute of Technology. Her field is chemistry. Her advisor is Heather Kulik, and her practicum was done at the SLAC National Linear Accelerator Laboratory.
So, hi everyone. Thanks for coming to my talk. Today I'll tell you a little bit about the work I've been doing for the last 5 years on large-scale analysis of electronic effects and protein structure. So first, why do we even care about proteins? So proteins are important to structure, function, and regulation in cells. And as Jared mentioned earlier today, we don't fully understand how they work. For instance, in enzymes, which catalyze chemical reactions, we cannot reproduce how proteins catalyze reactions so well. So there's something we're missing in our understanding of how proteins function. So one thing that's really important to proteins is their structure. So in the case of potassium channels, which allow potassium ions to pass through cellular membranes, there are dipoles along the inside of this channel that coordinate the potassium ion very specifically so that, for instance, sodium ions cannot pass through. You might have learned in high school the lock and key model of enzyme catalysis, where the enzyme is like a lock and the substrate goes in as if it's a key and it's perfectly coordinated, and then some function goes through. So chymotrypsin is an example of an enzyme that basically functions under this model. This so-called catalytic triad perfectly coordinates this, the substrate that goes in, and it cleaves a peptide bond. But this isn't true for all enzymes. For aliphatic halogenase, the reaction that it performs, which is halogenation, it sticks on halogen atoms to its substrate. This is a really reactive reaction. So in order to protect this active site from going out and adding halogens to lots of random things, the enzyme has protected this very deeply within the protein. And so in order to perform this function, its partner, and the partner has a long wiggly arm that burrows in to deliver the substrate. And so with the protein structure, that's a starting off point for understanding how this enzyme functions. The structure tells you some information. It's a good starting off point for simulation, but you also need to understand the dynamics. So structure guides function, but it's not the whole story. So to back up a little bit, a brief overview of protein structure. So you'll often see proteins drawn in this kind of cartoon model. But at its core, a protein is a long chain of amino acids. So every amino acid, or I might also call it a residue, has the same backbone. But there's 20 different possible side chain groups that have different properties. They might be positively charged or negatively charged. They might be polar. They might be nonpolar. This primary long chain of amino acids then folds up to form the protein's secondary structure. And the two common ones are the alpha helix on top and the beta sheet on the bottom. And you can see that some of those elements are present on the left. And these black lines between atoms that are drawn represent hydrogen bonds, which are really what hold a protein together. So a hydrogen bond occurs when there's an electronegative atom that is interacting primarily electrostatically with another hydrogen that's bonded to an electronegative atom. So electronegative atoms pull electron density towards themselves. So it has a slight negative charge. And the hydrogen atom that's bonded to the other electronegative atom will therefore be slightly positive. And so there's a charge interaction. And how do we get these protein structures? So experimentally, these can be determined by X-ray crystallography. There's other methods that have been used to determine protein structures, but I'll primarily be talking about X-ray crystal structures. So since X-ray crystallography has been an established method for many decades now, there's a huge repository of protein structures that have served as a source for unexpected chemistries. So for instance, there's this so-called carbon bond, which you chemically wouldn't really expect to be that favorable. But evidence for this, it shows up in high quality crystal structures. And later on, people did high-level simulations to understand why they formed. Even really ubiquitous interactions have been found by mining the protein DataBank. For instance, all amino acids have a backbone carbonyl. This carbonyl interacts with other parts of the protein in a very weak way. But because it's so ubiquitous, these ubiquitously weak interactions add up to have some impact. They're also a source for understanding unusual hydrogen bonds. So hydrogen bonds being very strong, a very strong form of non-covalent interaction, are a very rich source of trying to understand how proteins form their internal structure. So in this case, a proline is known as an alpha helix breaker because when it appears in an alpha helix, the alpha helix stops. And part of this might be because it forms this nitrogen-hydrogen-nitrogen-hydrogen bond to its adjacent residue. What kinds of unusual chemistry can we find in the protein DataBank? So one place we might look for unusual chemistries is where people think there's a mistake. So when X-ray crystallographer tries to develop a model of the protein structure, they have to fit a set of atom positions to the raw experimental data. And oftentimes, this isn't the experimental data doesn't tell you exactly where a specific atom is. So there needs to be some kind of chemical intuition added on top. And one of these sources of chemical intuition is atoms don't like to overlap. Their electron clouds are repulsive. So in this case on the left, there's a choice between the tautomer, so it can either be in this orientation or this orientation. And in the bottom, there's more overlap between the atoms. And so the correct way to orient it is this top one. And so clashes are a powerful tool for diagnosing local fitting problems and are one of the ways that people judge the quality of a protein structure. So if a protein structure has too many clashes, it's considered a poorly solved structure. But there's the case that made us think, wait, maybe this is a source of unusual chemistry. Maybe some of these clashes should really be there, is the case of catechol-O-methyltransferase. So this inactivates catecholamine neurotransmitters, which are things like dopamine. And in the active site, the catechol and the methionine, which donates this methyl group, are positioned closely. So closely that in around a dozen crystal structures, they all have a distance range from 2.5 to 2.75 angstroms. However, if we were to model this with standard methods used to model proteins, molecular mechanics, we never simulate this distance. The distance is much longer, around 3.5 angstroms. So how do we validate unusual interactions like these close contacts that we might not expect to be favorable? So in general, these unusually close contacts belong to a class of interactions of non-covalent interactions, which are notoriously difficult to model. So standardly, proteins are modeled with classical methods where the force fields are parameterized from experiment and previous computation. And in this particular data set, you can see that if the force field really was very good at reproducing the reference energies, they would all the dots would fall along the y equals x line, but quite a few fall off of it. And even in in quantum chemistry methods like DFT, they don't capture all of the contributors that are very important for accurately modeling non-covalent interactions like dispersion. So one of the questions that I would like to answer in my research is, how well do standard methods capture unusual interactions? When does it get it right, and when does it fail? So the overall structure of this project is, first, we're going to curate a data set of high-quality structures so that if we see an unusual interaction, like a close contact, in this data set, we kind of believe it more. We believe that the experiment is telling us that these actually exist, as opposed to it being an artifact of the experiment. We'll then talk about some minimal models to try and understand if these two residues are just in close contact, does molecular mechanics and quantum mechanics say if that is a supported interaction? And then we'll put it back into the protein environment as a whole. So first, how do we curate a data set? So the Protein DataBank is conveniently a repository of all solved protein structures. There's over 130,000 protein structures, which we probably can't analyze all of those. And more importantly, we don't want to analyze all of them, because the resolution of a protein structure tells you how precisely that atomic location is known. So if you look at the blobs on the top left, the electron density, you can see that you don't really know precisely where atomic locations are. So we really need to look only at high resolution data because that's what is experimentally supported. We also want to not look at the same protein over and over. And we also want to check for the quality of structure. So we want to assess essentially the quality of a fit between the model and the data. But we don't want to use metrics that exclude unusual features, like the clash score, because that will automatically exclude the features that we are interested in. So I define this close contact as 85% of the van der Waals radii. This will exclude ordinary hydrogen bonds, but capture unusually strong interactions like charge-assisted hydrogen bonds. When we look at what residues interact with what other residues, we can see if it fits our intuition. For instance, residues that are oppositely charged, we expect to interact very strongly because electrostatics. And we see that those are well represented. Additionally, we see that residues that can hydrogen bond to form this particularly strong non-covalent interaction are also well represented. In fact, tyrosine is extremely overrepresented based on its abundance in the protein data set. We also see unusual interactions. We see residues that are usually the same charge interacting, and we see nonpolar residues that don't have as much electrostatic interaction are also present. So instead of modeling the whole protein, we first model a minimal model of just 2 residues. And we can examine particular cases that might be interesting. For instance, those hydrophobic interactions, those hydrophobic residues, what are they interacting with? How are they interacting? So the ones that we expect would be most likely to be actually supported would be the ones where they might be interacting with the negatively charged residue. And we see that in these cases, the interactions that are favorable are the ones where they're potentially forming a carbon-hydrogen-oxygen bond. And where it's unfavorable is where that's not a possible mode of interaction. We might also be interested in the more expected hydrogen bonds from tyrosine. And we can see that in this case, when it's interacting with asparagine and glutamine, it has 2 possible hydrogen bonds. It can either be hydrogen bonding with oxygen or nitrogen. But we actually find that although the oxygen-hydrogen-oxygen bonds are more favorable than the nitrogen mode ones, that actually there's this unusual motif where it's forming 2 hydrogen bonds at once that is the most favorable and strongly interacting. It gives us some of the ideal orientation for one hydrogen bond, but it gains another hydrogen bond in return. So if we put the protein back into the protein environment by taking the close contact and a huge region around it and freezing all of the other residues, what happens to the close contact? So in this case, the 2 things that are in close contact are on the bottom. It's 2.6 angstrom, which is quite short. And there's another residue that's close by that's also interacting through form of hydrogen bond. If we do a QM optimization, we see that not much changes. It looks pretty similar. However, MM optimization does not find this other hydrogen bond, and so things have moved much more. So I want to conclude to briefly wrap up. We searched for short distances that could be considered mistakes in the protein data bank, and we found that these are quite ubiquitous. We found that some of these interactions could be motifs that haven't been previously described, like this double hydrogen bond. And large-scale electronic structure validates these close contacts when classical simulation doesn't necessarily. So I'd like to thank my advisor, Heather Kulik, and the lab for all their support. And also, of course, the CSGF for their funding and for all the friendships we've made over the last 5 years. Thank you.